Hello, and thank you for joining us. I'm Kristen McPike, Program and Alliance Manager at the Antibody Society. Today's scientific briefing is one in a series designed to inform and educate our members and the broader scientific community about topics related to antibody discovery and development. Our expert speaker, Dr. Francisco Ylera, will be discussing the landscape of phage display and therapeutic antibody discovery with us today. Please note this briefing is being recorded, and you can add any and all questions to the Q&A box or to the chat box in the viewer, and the questions will be answered at the end of the presentation. Without further ado, I'll turn the show over to our speaker.
Thank you, Kristen. Yes, so welcome to my webinar on phage display. In this webinar, I want to explain briefly what is phage display. Probably most of you are familiar with the basics. Then go into the advantages of phage display, the challenges, and then demonstrate what is possible if you use a modern technology and a modern library to generate what kind of antibodies you can generate. So phage display was invented in the late 80s, and it was the first technology that linked the phenotype, the protein here shown as a Fab, to its genotype, the DNA that encodes the Fab. And that was done by fusing the Fab to the phage code protein, the P3 protein, genetic fusion. And then the DNA is packaged inside the phage code. And so we have this linkage between the protein and the DNA, and this allows to select for proteins and peptides with specific functionalities, for example, binding to antigens or enzymatic activity and so on. So once it was discovered, it was quickly transferred to the antibody discovery field. Basically because at that time it was not possible to generate human antibodies. So antibodies came out of immunization and then had to be humanized afterwards. And this allows to generate human antibody libraries and to directly select for human antibodies. And so it was a big step at that time. And in 2018, it was awarded the Nobel Prize for George Smith and Greg Winter. So to make it clear in the beginning, the difference between selection and screening. So I use selection when we have a library that is so big that we cannot array it, so that we cannot test every single member separately as we do it in a screening. So in a screening, we have, let's say, a 384 well plate with candidates. We test it for binding, and then we know where the hits are. In selection, we start with a pool with a high diversity, and we end up with a high diversity just enriched in what we are selecting for. And for phage display, we need the selection step because diversity is so large. We usually speak of 10 to the 8 up to 10 to the 11 different, let's say, antibodies. And yeah, it's obvious that you cannot array such a large number. So what we do is we have our library shown here as a vial, and then we immobilize the antigen on a solid support. That could be an ELISA plate or beads. We add the library, and then we go through several rounds of selection and amplification. Most often 2 to 4 rounds are used. And in this step here, it is shown at the bottom. The antigen is immobilized. We add the library. All the Fab phages which do not bind to the antigens are washed away, and then the specific binding Fab phages are eluted. Then we come to the amplification step. So phages are actually viruses that infect bacteria. So we can take these phages to infect bacteria, let it grow overnight to amplify it, produce new phages, and then go into a new selection round. So we usually do 3 rounds. After 3 rounds, we take the DNA, subclone it into an expression vector, and then we come to the screening step. So we transform it into E. coli, pick separate individual colonies, and then screen for the required cross-reactivity profile. We send a number of hits for sequencing to identify which ones are actually unique antibodies, and then those are expressed, purified, and QC'd. And this process usually takes 8 weeks. So when we talk of antibody discovery, then phage display is used in different settings. One setting is that you can use it after immunization. If you shuffle the VH and VL libraries from the B cells or plasma cells, then through the combination, you end up with a large library again. So to find the real binders, it's a convenient method to use phage display so that you narrow it down to a handleable number of candidates, which then can be expressed and purified. But here in this presentation, I will focus on the second point, human antibodies. So with phage display, you can generate very large human antibody libraries, and I will focus mostly on that in on the rest of the talk. But it can also be used to generate variants of single candidates, for example, for affinity maturation. So you introduce new diversity into one parental antibody by, for example, exchanging CDRs, and then select for candidates with a higher affinity. Or humanization is also something where phage display is used to generate different mutants, humanized versions of your parental antibody, and then select for those that still bind with a good affinity to the target, so that the affinity doesn't get lost during the humanization process. Phage display is a very fast process. It's cost-effective, robust, and it's an animal-free in vitro process, so you also have happy mice in your lab. In vitro selection means that you have several new options which you do not have or not in that degree with animal-based methods. You can use toxic antigens, non-immunogenic or conserved antigens, and there's a very large target versatility that can be used with phage display, from small molecules to peptides, proteins, complexes, full viruses or cells, up to tissue sections. The process of the selection, we call it guided selection, and I will show you several examples for that on the next slides. You can select for epitope-specific antibodies, for mutation or post-translational modification-specific antibodies, or even for conformational antibodies. And since it's in vitro, you can control the selection conditions in terms of, for example, pH, salt, temperature, competitive binding, or for desired biophysical properties. Nowadays, not such a big topic anymore, but especially in the beginning, in the 90s, that was a very important aspect. After phage display, you have the antibody DNA on the sequence, so there's no clone loss anymore if you compare it to hybridoma technology. And of course, once you have the sequence and the DNA, you can very easily format it into other antibody formats, IgG, IgA, IgM, and so on, or the fragments, single chain, Fab, and so on. So that is simple subcloning. So let's look at some selections. Let's start with antibodies against a toxic antigen. For this, I chose an ADC, an antibody-drug conjugate, as an example, brentuximab vedotin. So we use the ADC as an antigen for the selection, and we also used MMAE. MMAE is the payload conjugated to BSA, and we want to have antibodies that specifically bind to the MMAE, not to the antibody. So either on the antibody or on the BSA, but not to the antibody by itself. And here you see the ELISA results for the outcome. In orange is the ADC, in blue is the BSA payload, and you can see that it binds nicely to the both proteins, both conjugates, but not to other unrelated proteins. And then we did a titration ELISA. We took one antibody against MMAE as a capture antibody, titrated the ADC, and detected with another anti-MMAE antibody conjugated to HRP. And you can see that we get a nice titration curve with a good sensitivity. So the methods we do it is that we immobilize the antigen, as mentioned before, and we can add a blocking antigen, what we call a closely related antigen, CIA, in excess to the phage library. So all antibodies that bind to the closely related antigen will be washed away, and we only get antibodies that bind specifically to the antigen. So in this example, we used it for the generation of anti-idiotypic antibodies. So the drug antibody is our target, immobilized here on the surface, and the closely related antigen is an isotype control IgG and human serum. So we have plenty of different IgGs in solution, and only the drug antibody is immobilized. So everything that binds to, let's say, the Fc domain will be washed away, and we get really paratope-specific antibodies out of this selection. And that works really nicely, so there is no need to screen for thousands of clones. You really get specifically the paratope-specific antibodies. And here's an example. We distinguish between three different types. Type 1 are the inhibitory binding anti-IDs. They bind to the paratope of the drug antibody, so they only detect the free drug when it's not target bound, because the antibody competes with the drug target, the same binding site, the same paratope of the drug. Shown here at the bottom for golimumab. So we have several antibodies with similar isotypes or binding to TNF-alpha, which are not recognized. We get a nice bar for golimumab, and we don't get a bar for the complex TNF-alpha and golimumab, because, as I said, it's inhibitory, so either the antibody binds or the target. The type 2 antibodies are non-inhibitory, so those antibodies can bind no matter whether the drug target is bound by the drug by the antibody or not. So here we can detect the free antibody, the partially bound antibody with one drug, or the fully bound antibody with two drugs. And you can see that here we have the green bar for golimumab and the blue one for the complex. But we don't see binding to TNF-alpha alone or to the other antibodies. And the type 3 antibodies are antibodies against the target antibody complex. So those antibodies do not bind to golimumab by itself, so no green bar here. They don't bind to TNF-alpha. They only bind to the complex, the blue bar here, TNF-alpha golimumab. And yeah, that's something that can be generated quite easily with phage display. Of course, you can also generate these antibodies through immunization and other methods, but there you often have to screen large numbers to actually distinguish between the Fc binders and then other binders and really pick out those antibodies with the specificity you need. So this way we actually generated a large number of anti-therapeutic antibodies which are in our catalog. Here's an example of the list. They are all highly specific, fully human, tested in PK and ADA assays, of course with a defined sequence, so secure supply, recombinant production, stringent batch-to-batch control. So if that is something of interest, then please visit our catalog and see whether we have the target you need already. So let's come to another example, the neutralization of West Nile virus proteinase, NS2B, NS3. So that's the enzyme, and the active site is known. That's the histidine 51 here. And we want to have antibodies that specifically blocks this enzymatic site. And the way we achieved this is that we used this NS2B, NS3 as our antigen. And we used a mutant of this enzyme for blocking, as a closely related antigen. And here we mutated the histidine at position 51 to an alanine. So all antibodies that bind up here, for example, can also bind to the mutant and will be washed away. And the only difference between the blocking antigen and our immobilized antigen is here at this position 51. So we get antibodies that bind here to the active site. And that's shown here for the titration of 3 antibodies that they nicely neutralize the activity of this protease. And yeah, that's data that is published. Of course, I can show only data here that has been published, cannot show customer data. And yeah, you will see that all the data is in-house data because it's simply easier to present this than searching through the literature for nice examples. With over 20 years of experience, we have a lot of nice examples we can show here. The next example is even, the difference is even smaller. It's not an amino acid, it's just a side group. It's the methyl group in thymidine replaced to this bromium in BRDU. These antibodies are used to monitor cell proliferation by incorporation of BRDU, and to do so you need antibodies that specifically bind to BRDU and are not cross-reactive to thymidine. Yeah, we used 3 different biotinylated DNA oligos with BRDU built in for the selection. So 3 selection rounds, 1 oligo per round, and use the same oligos with a thymidine for blocking. And this way we could generate antibodies that show a nice shift here in flow when they proliferate. We do not see, or we do not see the shift, or hardly a shift, if we use chlor instead of bromium. The same for iodo DU. With thymidine treated, we don't see a shift at all. So there's a very nice difference. And also in immunofluorescence, this antibody can be used to nicely stain the proliferating cells. And I have another example for a truncated protein. So here we want to distinguish between CXCL12, the full-length version, 1 to 77, and the truncated version, 3 to 77. So obviously from 3 to 77, both proteins are the same. So it's just the N-terminus which is different here. Yeah, we managed to generate antibodies that are specific for the truncated version, shown in the upper figure, as well as for the full-length version, which do not bind to the truncated version. So here in the upper assay, the antibodies against the truncated version were used as capture antibodies, and then some other CXCL10 antibodies for detection. And at the bottom example, antibodies that specifically bind to the full-length version were used as capture antibodies, and then antibodies which bind to the later stretch for detection. And these antibodies are actually used in an CLAI certified assay. So it's not just an academic task, but really there are applications behind it. I mentioned that we have 3 panning rounds that can also be used to generate antibodies that bind to 2 or 3 different antigens that can have a certain sequence homology. A typical example is that we generate antibodies against the human target and the mouse or cyno homolog protein. And that's done by using the human antigen in the first round, the mouse antigen in the second round, and then the human again in the third round. And this way we get antibodies which bind to both forms similarly. And I will show an example on one of the next slides. First, I want to come to antibodies with complex binding profile. So as I said, the whole process is in vitro, so we have the option to modify the ionic strength, for example. We can add or leave out certain ions, for example, calcium. We can adjust the pH according to the requirements. For example, binding at a certain pH or elution at a certain pH or ionic strength. Or we can do the selection in the presence of cofactors. And the example I want to show is for an oncogen. It's antibodies that should bind to a tumor target in the acidic microtumor environment at pH 6, but not to healthy cells at pH 7.4. So here in the ELISA, you see in dark green the human protein, in light green the cyanoprotein, both at pH 6. So you see nice binding at pH 6 with similar signal strengths on both species. And in blue, dark blue is the human, in light blue is the cyno protein at pH 7.4. So you see some slight cross-reactivity for 2 candidates, but all the others are really specific, bind only at pH 6. And here at the bottom, we have a titration in flow cytometry on the target on cells. And you can see in light blue the titration at pH 6. So we have a nice binding curve, whereas at pH 7.4 the line stays flat, so no binding to cells at neutral pH, and nice binding concentration dependent at pH 6. But we can also do the opposite. We have selected antibodies against IgM that are used for affinity chromatography. And here we want that the IgM binds to the immobilized antibody on the resin at neutral pH, but we want to elute the IgM at very mild conditions at pH 5. And yeah, we generated antibodies that allow exactly this. Okay, enough examples about specificity. Let's look at some antibody properties. Let's start with affinity. So first of all, to select high affinity antibodies, a monovalent display is very essential. Um, for those of you who are not familiar with affinity and avidity, the binding of the monovalent antibody to its target, it's called the affinity. But if you have two arms, like in an IgG, or if you have several copies of a Fab on the surface of the phage, then we speak of avidity. So the antibody binds much tighter than it would if it would have only one arm. So, for example, if you have an antibody with a monovalent affinity of 10 nanomolar and uh, probably 500 picomolar, then in a bivalent format, you cannot really distinguish between these two anymore because with the washing conditions during phage display, they both show similar binding properties, and the high affinity candidate would not have an advantage anymore. And so if you don't have a monovalent display, then you cannot really select for high affinity, and the resulting antibodies are all um, good to medium affinity. So that is one very important aspect. The second aspect is the library size, and that's shown here in the diagram. On the x-axis is the library diversity, the library size plotted, and on the y-axis, the affinity of the best antibody reported from this library in the literature. And you can see as the libraries became bigger, also the affinities, the reported affinities became better and better. Um, so if you have heard that phage display antibodies don't have a good affinity, then it's basically because in the 90s, when the method came out, um, the focus was more on generating human libraries, but it was not so well known in the beginning how big a library has to be. And yeah, these essential display parameters were not that easy to control. And this way, it was possible to select antibodies, but many antibodies were not of such great affinity. But with a well-designed modern library, it is possible to deliver antibodies in the low picomolar range. And I will show you some more data on that in a minute. Actually, here I would like to introduce you to our Pioneer library, which we have launched about 3 years ago. It's a synthetic human naive Fab library. It is highly curated for sequences that are ideally suited for phage display and expression in E. coli, because if they are not well expressed in E. coli, then they will be lost during the selection process. We used germline genes with known beneficial properties that are also frequently found in therapeutic antibodies. So we took two heavy chains, VH1 and VH3, and two light chains, kappa and lambda. And this way we can generate 4 sublibraries, VH1 kappa, VH1 lambda, VH3 kappa, and VH3 lambda. We have a germline-specific CDR design with all 6 CDRs diversified. We have strongly reduced potential post-translational modification sites and other liability motives. We have the SpyTag built in. I will explain more about the SpyTag on the next slide. And the Pioneer library is one of the largest, if not the largest, functional antibody phage display library ever made, with more than 200 billion unique antibodies. But first, let's come to the SpyTag. So the SpyTag technology was developed in the Howard lab at the Oxford University. It consists of a small peptide tag, the SpyTag, and a small protein, the SpyCatcher. And if you mix these two, then they spontaneously, autocatalytically form an isopeptide bond between a lysine side chain and an aspartic acid side chain, but not between random ones only between certain ones in the SpyTag and in the SpyCatcher. It's a very specific reaction. It's a covalent bond that is formed. It's irreversibly formed. And this reaction is very fast and quantitative. So we used it in our library. We added the SpyTag to our Fabs, and we cloned the library into the expression vector right away. So our vector is the expression vector. It doesn't have any other phage display elements. And we use an E. coli strain with a modified genome that encodes SpyCatcher P3 fusion protein. And then the SpyCatcher P3 fusion and the Fab with the SpyTag can ligate within the E. coli, and we get a covalently displayed Fab on the surface of the phage. And the advantages of SpyDisplays are, first of all, the Fab is expressed without large fusion proteins, so it's smaller, it can fold nicely. It's a very rapid protocol. We have one panning round per day, and we have no subcloning step between selection and expression because the library is already in the expression vector. And also very important, we have a true monovalent display. That means we have a maximum of one Fab per phage. Yeah, that's, as I explained already, is very essential for the selection of high-affinity antibodies. So in summary, with SpyDisplay, we can very fast and efficiently select for high-affinity antibodies. And here's an example for selections against 6 different targets. You can see here the affinities of the analyzed antibodies against the various antigens. And you can see that we have a good number of antibodies in the sub-nanomolar range down to the low picomolar range. So in total, about 25% of the analyzed clones have affinities in the sub-nanomolar range, and 2/3 of the antibodies are better than 10 nanomolar. In case you are not familiar with these orders, most therapeutic antibodies are in a range, I would say, 5 nanomolar and better. Yeah, usually it's not that easy to get antibodies in the sub-nanomolar range because the higher the affinity, the more difficult to get to the antibody generally with every method. When we look at developability, then it's important to mention first that this is a parameter which is not looked at because of phage display antibodies. It's the same for antibodies from immunization. Before they go into clinic, they usually undergo developability optimization to remove sequence liabilities, for example, motifs that are prone to aggregation, degradation, or immunogenicity, PTM sites, or like glycosylation, deamidation, oxidation sites. Some antibodies are simply difficult to produce in CHO cells and other expression systems. And some antibodies just don't behave well if they are concentrated to very high concentrations, like 100 mg per ml, what is needed for subcutaneous delivery. Then the viscosity might be too high, or they tend to form oligomers. So these are the developability, or some of the developability parameters which always have to be looked at. In the beginning, in the 90s and also early 2000s, that was a time when the therapeutic antibody boom started. People were happy to have a specific binding antibody, and then after the first failures in the clinic, it became more and more obvious that developability is very important characteristic of the antibodies and should be looked at the sooner the better. But of course, with phage display, the focus at that time was more on how to optimize the libraries, the selection conditions, and not so much the developability. I mean, not only with phage display antibodies, but some antibodies with poor developability basically spoiled the reputation of phage display. However, if you have a modern library, then the developability can be built into the library already, so that you get actually good antibodies directly out of your selection, and no further optimization is ideally required. And here on the right, I show different motives, liability motives. In green is the number of, or the percentage of antibodies affected in the Pioneer library. In blue, the human antibody repertoire. And in red, the antibodies that are under development in the clinic or already on the market. And you can see that the green bar is usually the shortest bar among these, or at least of similar size at the most. Since we used germline genes with known beneficial properties, we get antibodies that already have good biophysical properties and a good expression profile. And yeah, as I just explained, reduced motives that could lead to concern later. And I will show, here it is already. We generated antibodies against 2 targets as what we call showcase projects, and compared these antibodies for their functionality and developability to antibodies in the clinic for TIGIT, domvanalimab, tiragolumab, and vibostolimab, and to 2 antibodies on the market for IL-6 receptor, cerulumab and tocilizumab. The greener the cell, the better the value. Red is poor value, white in the middle. And you can see that we get antibodies with good affinities, as good or better than the benchmark antibodies. Functionality in a reporter cell assay, again, similar or better than the benchmarks. We have a good thermal stability, good monodispersity, a low self-interaction propensity measured by SI-BLI. Hydrophobicity is, for most of these antibodies, very good, measured by Hick. There are some candidates which are not perfect yet. The antibodies are all stable in an accelerated stability study and passed our polyreactivity test. So you can see that it is actually possible to generate antibodies with very good properties directly out of the library without any optimization, no affinity maturation, no developability optimization, whereas the antibodies on the market or in the clinic, probably, I don't know it for these candidates, underwent optimization before they could enter the clinic. So that is the developability conclusion. Excellent affinities, functionality, high thermal stability, low hydrophobicity, and self-interaction propensity, comparable or even better than antibodies under development. And with that, I would like to come to some challenging projects against GPCRs, a class that is known to be difficult to generate antibodies against. And here we have 2 projects that I want to show in collaboration with 2 Swedish companies. We start with antibodies against C5 receptor. Here we used approach, this is a cell panning approach. So we used C5 receptor overexpressing cells from SWA. And we selected a good number of antibodies and then tested these antibodies in a reporter cell assay using the islet cells from SWA. That's shown here. In red and in black are the two benchmark antibodies, izastobart and avdoralimab. And the other colors are for the Pioneer antibodies. And for the green one, for example, you can see that it has a better functionality than these benchmarks. And here at the bottom, we have the table again with all the parameters. We don't have the purified protein, so we did a titration by flow cytometry to, and here's the EC50 value. So we have good EC50 values, better than the benchmark antibodies, good functionality in this reporter cell assay, and also the other developability parameters are quite nice. So again, even for these challenging targets, we got antibodies with a high affinity and with a good functionality and developability profile. In the second example, we generated antibodies against CXCR4, and here we used the purified protein stabilized with scaffold protein from Salipro Biotech, a company that is specialized in stabilizing transmembrane proteins. Here you see the 3D structure of CXCR4 stabilized with this scaffold. So this was then used for normal protein panning. We got a good number of antibodies. Then we tested for functionality in a chemotaxis transwell migration assay. So the number of cells that migrate if no antibody is present or if an isotype control antibody is present is set to 100%. And if we add the benchmark antibody, ulocuplumab, from the clinic, then we see that only 10% of the cells can still migrate to the ligand CXCL12. And with our Pioneer antibodies, we are in a similar or even slightly better range than this benchmark antibodies. And again here, the green table showing that we have good affinity. Here we have the protein, so we could measure affinities. Yeah, good functionality in this migration assay and also good developability parameters. I mentioned in the beginning already that Phage display is very fast, but for the Pioneer platform, we had especially speed in mind. So it's not just a library, we call it an antibody discovery platform because it consists of different technologies. First of all, the library itself, but also SpyDisplay, what I mentioned already. Just as a reminder, there's a covalent display on the surface via the SpyTag, SpyCatcher protein ligation. Then obviously all the resulting antibodies carry a SpyTag. So they are compatible with our modular antibody assembly platform, Trailblazer. So once the Fab with the SpyTag is purified, we can mix it with one of these catches, which we all have sitting in our freezer or are available through our catalog. And within 1 hour, the protein ligation is complete, and all the antibodies carry the SpyCatcher. In this example here, site-specifically conjugated SpyCatcher without modification of the binding site. So we see better performance in assays than conventionally labeled antibodies. And of course, it is much easier and faster. But we can also convert all the antibodies in this into the synthetic IgG format. And here we have all kinds of Fc catchers, IgG1, 2, 3, 4, IgA, IgM. So we can take the whole output and within 1 hour convert it into the synthetic IgG format and continue our screening and characterization. So it's not the final drug format. It's just prototype for accelerating the characterization of the antibodies before we then, for most projects, go into the IgG format. And the last technology, I will not go into details here in this webinar, is our Spylock technology to generate bispecific prototypes in high throughput very rapidly. So we can generate hundreds of these bispecific molecules basically in a day for screening which are the best antibody pairs. And with this, I want to come to the summary. So I showed you that with a modern, well-designed library, it is possible to generate a large number of antibodies, get a high diversity. I haven't shown the epitope coverage, but we also have data for that it is actually very good for Pioneer antibodies. We have excellent affinities and functionality, good developability right out of the library, and the method is fast. Within 4 weeks, we can generate the purified antibodies. That's probably we are one of the fastest methods for antibody generation available right now. And as I showed, the candidates compare very favorably to antibodies in the clinic or on the market. And with that, I want to say thank you, and I'm happy to answer your questions.
Thank you so much, Dr. Ylera, for this wonderful, engaging conversation. And also thank you to the audience who has added so many questions to our Q&A. So I will kick that off. First question is from Tong Yang, who says, how was your library generated, protein or cell immunization? And what's your opinion on synthesis nanobody sequences in the target validation?
So how is the library generated? It's basically it's based on a consensus approach on human antibodies. It's a naive library, so not an immune library. And more details are actually published. Last year in summer in MAPS, we published the more data on the Pioneer library, so it can all be read. The second question, what was it? Nanobody libraries?
What's your opinion on synthesis nanobody sequences in the target validation?
Yeah, nanobodies are actually a newer class which gained a lot of attention in the last, I don't know, 5 to 10 years. Both for target validation and tool antibodies as well as for therapeutic antibodies. For therapeutic antibodies, I believe it is fueled a lot through the high interest in bispecific antibodies, because one chain makes it much easier to generate bispecifics without having the light chain heavy chain pairing issues. We don't offer VHH or nanobody library, so yeah, I think I don't want to discuss here now the pros and cons of nanobodies, which are definitely there, biophysical properties of humanized versions and so on. But I think there are others who can do that better.
That absolutely makes sense. And a follow-up question from Adrian Sachs. Have you had any experience with canine phage display library? Anything specific to be aware of different from humans?
No, we don't have any canine libraries. Those exist. There are companies that have generated such libraries and generate antibodies to treat dogs. But again, I don't have the details, the insight on those. We focus on human antibodies.
Thank you so much. The next question comes from William Clark. Are you still using the Morphosys library? And then there are a couple comments. Their last generation had optimized frameworks. Are you allowed to use this one? Now that Novartis owns Morphosys, will this library still be allowed to be licensed?
So the last question first, whether it can be licensed. You have to contact Novartis. I don't know. And whether we still use these libraries, yes. So we use the UCAL libraries for more than 2 decades, and we still have them in our labs, the UCAL Platinum library. I mean, we started with the gold and then switched to the UCAL Platinum. We don't use the 𝗔𝗯cie library with certain framework genes. We don't have a license for this one. And the UCAL libraries we can only use for tool antibodies, for research and diagnostics, not for therapeutic applications. And that was also one reason why we decided to generate a new library, the Pioneer library, but also yeah, we thought it's time to come up with something new that's we thought we can do it better, and I think the data we generated for the Pioneer Antibody Library shows that it has many features that are yeah, excellent.
Super exciting innovations we're discussing here. Next question comes from Alan Engemelmo. And Alan says, it is reported an unexpected frame shifts from gene to express protein in a phage displayed peptide library from Caracamo, Rivera, and Brissett et al. From 1998. And Alan linked that article in the chat if you're curious. Could you comment on this based on your experience? Have you ever faced this issue with phage display libraries?
Yeah, frame shifts can happen during the cloning, during the synthesis of the DNA, depending on how you do it. And it's just an artifact that yeah, happens during the library preparation. So that is the same with the Pioneer library. So what we did is for the light chain and for the heavy chain separately, we had a selection step to remove genes with a frame shift, so that only antibodies that are cloned without any frame shifts and insertions or deletions um, are um, uh, selected and end up in the final library beta-lactamase selection step. And yeah, we saw during the QC of the library that it actually worked very well. So if the question aims at truncated clones, which are enriched because they are shorter DNA, shorter proteins, better expression profile, that's something we have seen in the past, not at a huge extent, but we haven't actually seen that for the Pioneer library yet. Probably because we could properly remove it from the library through this selection step, but also for the UCAL libraries, it's a minor problem.
Understood. Thank you so much. Next question comes from Kieran Pitchers. Is there any advantage over pre-blocking the phage in solution with the CRA compared to subtracting the non-specific phage with immobilized CRA before panning against your antigen?
Yeah, it's both possible. You can do a pre-adsorption on the CIA or post-adsorption. We feel or we see that the addition to the library in solution is more stringent than pre and post-absorption.
Thank you so much. And another question from William Clark. Anyone using your library for snake venoms, a lot of antitoxins that are horse polyclonals, a consistent supply would be beneficial.
Yeah, we haven't done such projects internally, but yeah, basically it would be possible. So if he's interested in such projects, then please contact us and we can discuss the details, how to set up such a project best. And yeah, supply, of course, is not an issue. As I explained, we have the sequence, so there's no clone loss, and the antibodies can be produced again and again in whatever scale necessary.
Thank you. Next question comes from Praise Aguntunkun. You talked briefly about selecting pH-sensitive antibodies. Can you describe how these libraries were designed and the antibodies selected?
Yeah, library design, it's our Pioneer library, so there's no difference in the library design. And the selection, there basically we performed the binding at the required pH, so pH 6, and we tested different. Oh, light went off here. Okay, we tested different blocking and elution steps. Yeah, actually, they different pannings were successful. So, for example, pre-adsorption at 1 pH, and then at neutral pH, and then transferring the supernatant to the antigen beads at pH 6. Yeah.
Great. Thank you. And now from 𝗔𝗯ance 𝗔𝗯ance, a concern regarding synthetic libraries is mostly off-target binding. Is this something you still observe, or do you believe this is something of the past for synthetic libraries?
It is a topic for all antibodies, not just phage display antibodies, and should be analyzed. We don't see un or a worrisome high rate of unspecific binding, as shown here in these green tables. And these are not just a few antibodies which do not show unspecific binding from many. So the percentage of unspecific binders is quite low. I don't have a number, but that's our experience so far. Probably also dependent on the library design.
Understood. Then William Clark, which thank you so much, William, for your engaging questions. How do you elute the SpyTag antibodies?
We have an protease cleavage site built in between the code protein and the spy catcher. So we can use an protease cleavage step to elute the phages, so the antibody stays bound on the antigen on the plate, and the phages are eluted and then used for infecting bacteria.
Then just a couple more. One from Sophie Gabriels. On the developability parameters, all tested clones pass the polyreactivity test. How is this determined? And how often do you observe polyreactivity for other clones? Finally, is this a synthetic library more prone to enriching for aspecific binders?
Let's start with the last question. We don't observe it, that the synthetic library is more prone for unspecific binders. The binding was tested in ELISA by binding to different antigens. Was it these two questions, or was there something else?
The how was the developability parameters, how were they determined? And or how was the polyreactivity test determined?
Yeah, that was measured by ELISA, measuring binding to different known antigens. Details are also in the publication.
Thank you. And Mao Lee shared the publication in the chat if anyone is interested in that. Um, Finally, Tong Yang also wanted to know, do you offer protein production service? And I think that Tong can follow up with you offline about those services from there. So I think that that's going to wrap up the questions. Thank you so much, Dr. Ylera, and thank you so much, audience, for such engaging and proactive interaction with us. People are also asking if the presentation is going to be available online at some point, and I will say from the Antibody Society end, we will be sharing this on YouTube, our YouTube channel, and anyone who registered should also get the recording. Dr. Ylera, will you be sharing the presentation separately, or is that something that we could do?
We could, yes.
Okay, perfect. So we will make sure that everyone who is registered will get a copy of the presentation. And we thank you all so much for attending.